This is Mutilda. Mutilda was a cow that a little while ago was walking around, mooing, having a good time, and gossiping with their friends about that new bull that moved into the nearby pasture. Recently, however, their appointment with the Reaper came due, and it was time for Mutilda to move on. And after a magical trip through the abattoir's portal door and some off-screen carnage, Mutilda was converted into a pile of steaks. But with a piece of her sitting in front of me, I have to wonder, is she really dead? She may not moo anymore, but does that mean she will never moo again? And when you eat a steak like this, is it really dead? This is what we're going to find out today. Throughout the video, we're going to explore the whole Frankenstein tech tree, and see about giving Mutilda a second chance at living once more. We've consulted all the most magical books, and every necromancy text we could find, including the most powerful of them all, Essential Cell Biology. But before we do that, put on your best 90s goth outfit and some heavy black makeup, because we need to talk about your many deaths. Spooky season has come early this year, and it turns out that you don't actually die once. You die a whole bunch of times. Your first death is the obvious one. Something has happened to disrupt the function of your brain. This could be because one or many of your other organs failed, or something happened to your brain itself. Maybe it was that sixth corn dog finally clogging an important artery, or maybe it was a Home Alone-style trap that is far more lethal in reality than in movie world. No matter the case, this is the least dead that you'll be moving forward.
I've seen worse.
Though, just because you've suffered terminal damage doesn't mean you're dead yet. Even just reaching this first stage of death can take a while. Unlike in video games, getting shot in the knee doesn't make you drop like a wet rag. And swords, while very lethal, are also not very quick killers in most cases. The human body is amazingly resilient to damage that ought to be fatal, so even if you will succumb eventually, it can take time. And on the list of bad ways to die are all the ones that involve it taking a while. For example, during the French Revolution, when they were inspecting the aristocrats to see if they were made out of cake, the removed heads were said to behave fairly normally for up to a few minutes, some looking around or trying to speak or gesture. Or, in the American Civil War, soldiers were left on the field for literally days, mortally wounded, unable to move, but still very alive. And the ones that did survive reported a strange glow coming from their wounds. Turns out that there was a naturally antibiotic-producing microbe that lived in the dirt they were dying in. It didn't invade their bodies, it just kept growing enough to keep the other microbes out. And these bugs happen to be naturally bioluminescent. They called it the Angel's Glow. Very spooky. But again, weirdly survivable. There are many cases of people experiencing some kind of intense trauma, be it hypothermia, poison, or otherwise, where they seem pretty dead on first inspection, but warm them up or let their liver slowly metabolize the poison, and up they pop. There are multiple reports of people being basically frozen solid due to an accident in the evening, and then not being found until the next day. And when the blue bodies were slowly warmed up, the victims woke back up, most making a full recovery eventually. And there have been legends and reports for ages of people consuming pufferfish, or were otherwise poisoned by tetrodotoxin, and then entered what looks, for all intents and purposes, like death. Heart not beating, body not moving, but suddenly, after a day, they've processed the poison and start to wake back up. This was even a central premise in one of my favorite anime called The Apothecary Diaries. It's also the origin of the concept of zombies. Zombies originally were supposedly the product of voodoo shaman. They would allegedly drug their victim with a mixture of tetrodotoxin and scopolamine to induce a death-like trance, allowing the shamans to steal the bodies and gain indentured servants.
Allegedly.
Of course, there's very little evidence of this working successfully, but it did spawn an entire genre of books, movies, and video games. And those are just the rare cases. If we want examples of people being brought back from the dead on the regular, we need only look at the first level of necromancy, which is, of course, modern medicine. Our hospitals are full of scalpel junkies that will do their best to kill you while the anesthesiologist keeps you alive. And they can take a pile of hamburger and stitch it back into Humpty Dumpty just so long as the brain survived. Depending on the exact damage, even if the world goes black and you think you're cooked, if you get to a doctor in time, they might be able to undead you. Unclog that artery, give you some fresh blood, or get your heart beating again, and you might be back in action.
Look who knows so much, huh? Well, it just so happens that your friend here is only mostly dead. There's a big difference between mostly dead and all dead. Please open his mouth.
One of the quirks of modern medicine, though, is that even if you do get to a doctor and your body lives, you might still be dead. I'm speaking of the you with a capital Y in this case, the one that lives in your brain. I've always viewed the body as just a big meat car that the brain drives around, and your soul is just the sum of electrical and biochemical activity in your brain. Your soul can't go anywhere. It's not a substance. It's an emergent property of a complex system. It's a series of memories, intuitions, learned behaviors, and genetics. If we look towards projects like the Blue Brain Project, where they are nearly at the point of simulating an entire mouse brain on a computer, we can get a glimpse into one potential avenue that necromancy may take one day. While it's too energy and resource intensive today, there may come a time not too far from now when running a human mind on a computer becomes possible. And at that point, you would become what is called substrate independent, no longer bound to your meat car. And maybe we get so good at growing brains that someday we can just take that data and build you a new brain to live in. But what if you're not scanned in time? Well, there's a small body of evidence that some memories might be stored in the form of nucleic acids, either RNA or through epigenetic changes. A set of snails were trained to respond to an electrical shock, and then once trained, were blended, and their RNA was extracted. That RNA was then given to naive snails, and the ones exposed to RNA from the pre-trained animals began exhibiting the same behavior. So maybe iZombie had it right, and eating brains gives you some of the memories. Alternatively, if we could map memories to the epigenetic changes they elicit in neurons, we might be able to piece together some scraps of your memory. There is also a long history going back more than 100 years of surgeons trying to transplant heads, and while they've managed a few partial successes, none of the animals that it's been attempted on have survived for very long. So at least for now, you get one copy of your soul, and if anything happens to it, the you with a capital Y is gone. Though that does bring up an important point. If modern medicine is the first level of necromancy, that does imply that there's actually a higher level. Let's call it necromancy level 0.5. As the expression goes, an ounce of prevention is worth a pound of cure. So if you want to avoid a trip to the local necromancers, your best bet is to take care of the body you already have. And since we are living machines, the easiest way to take care of your body is by choosing what goes in your food hole. Eating healthy isn't always easy, though. I know when I get busy, especially when a video is nearly due, I really struggle with eating properly. Taking the time to cook isn't always an option, and often the readily available options are junk. But that's changing thanks to the sponsor of this video, Factor. Factor is the amazing subscription service that delivers ready-to-eat meals right to your door. Once a week, you'll receive a box of pre-cooked, chilled meals that are absolutely delicious. There is a massive selection, and it changes each week, so you always have fresh choices. All you have to do is warm them up when you get hungry. And if you have specific diet needs, like you're vegetarian or doing keto, they have great options to choose from. The first one I tried was beef mushrooms and gravy on mashed potatoes with a side of roast carrots and squash. It was really great, well seasoned, healthy, and delicious, and it only took moments to warm up. Honestly, this is going to make both editing and doing science so much nicer. Now I can have a proper nutritious meal without having to break my concentration or stop what I'm doing. And the diversity of options is great, so me and the crew will be eating a lot better from now on. Speaking of which, I also had my crew member Frank, try one.
Oh yeah.
Good?
Yeah, this is actually really nice.
So if you want ready-to-go healthy meals delivered right to your door, head to factor75.com and use the coupon code ThoughtEmporiumFB50 to get 50% off and free breakfast for a whole year. That's code ThoughtEmporiumFB50 and factor75.com for 50% off and free breakfast for a whole year. Now, with our bellies full and our bodies repairing themselves as they should be, we can return to our goth descent into the gloom and continue our discussion of your many deaths. So, what happens when the car survives, but the driver doesn't? Brain death is when the brain can't be rebooted. It's an all-out collapse of the coordination of the brain, and the cellular activity drops immensely. And while I contend there may be ways to even reverse this someday, at least today, this is it, Chief. At this point, you're cooked.
He's dead, Jim.
However, with the help of a heart and lung machine, IV fluids, and frankly a legion of attendants, modern medicine can hold your body in this state for an incredible amount of time, potentially a decade or more. But if your soul is just the sum of the neural activity in your brain, the body in that hospital bed is, for all intents and purposes, alive but soulless. A shell that keeps fixing itself with nothing to drive it around. At some point, though, when the support is gone, the body can start its rapid descent into... This is, quite literally, the meat of this video. Death number 2 is cell death. It's easy to fall into the trap of thinking that as soon as you pull the plug, all the cells die quickly from lack of oxygen. But is that really what happens? From the moment this process starts, cells begin to die, but the shape of that die-off curve makes a huge difference. In video games, the curve is a cliff. 100% death instantly from an arrow in the knee. But how steep is the curve in reality? And how long can individual cells hang on? Well, the specifics depend greatly on what organ and tissue type we're talking about, and also importantly, how you died. If you succumb to disease... honestly, you've been rotting in that bed for a while anyway, so things are going to break down a lot faster. But assuming the body was healthy, and for example, you're one of the aristocrats that were found not to be made of cake, the body shuts down remarkably slowly. These days, it's routine to transplant an organ even an hour or two after the organ was harvested. Just keep it cold in your lunchbox and you can fly it in a helicopter across town. So cells are fine for an hour or two with no blood flow or fresh oxygen. Recently, we started to see heart-in-a-box technology be used, where donor organs can be kept fully functional for 12 hours. They give it a blood supply and oxygenate it, and then, of course, keep it beating. And half a day later, somebody new gets to keep it beating in their chest. Truly, the biggest impediment to a proper Frankenstein attempt these days isn't the mechanical aspects of connecting up body parts plumbing. If they're fresh enough, the scalpel jockeys could probably do it no problem. It's actually more of a problem with transplant rejection due to tissue mismatch, and, importantly, connecting up the nerves, which is famously difficult. But those organs and tissues are, by all normal definitions, fully alive at the time of transplant, and were carefully tended when they were harvested specifically so they wouldn't die. What if you don't tend them, and instead we're just talking about a corpse in the morgue? Well, in the 1960s, to study this question, they took a liver and left it out on a desk at room temperature to see how its activity decayed over time. And while, yes, obviously a lot of function was gone by the next day, it still retained an astonishing 12 to 16% activity. Which, look, that isn't a lot, certainly not enough to transplant, but it's a lot more than you might expect for a liver that's been sat out like bad potpourri. And this was in the 1960s when they were using radioactive tracers to measure this. These days, bone marrow is recoverable and usable even up to 5 days after a body dies, potentially longer if it's kept cold. With no blood flow, the core of a bone has exactly 0 fresh oxygen coming in, and if it's usable at 5 days, it's alive for a lot longer than that. It just stops being medically useful as you're not going to get a usable amount of cells for transplant. There is, however, a key point on the die-off curve that's important to today's discussion, and that is the point where you can no longer extract a tissue sample that has enough live cells left to establish a culture of. In the lab, we routinely use cells that were taken as either a biopsy from a live animal or a sample from an animal that was sacrificed immediately before harvesting the tissue. And these cells, thanks to our careful tending, can outlive the source animal for a very long time. I mean, doubly so if you blended a mouse to get those cells in the first place. One extremely famous example is that of Henrietta Lacks. She was an American woman that died in 1950 around the age of 30 from a rather aggressive cancer. But not before her doctors removed a part of the tumor and managed to get it growing in cell culture. This was a huge moment because growing animal cells had been fiendishly difficult before then. However, being doctors in the 1950s, and of course Henrietta being black, meant that they didn't bother to consult her on the use of the tissue or pay any kind of compensation for the research breakthrough that she was an intimate part of. The scientists just kept splitting the cells, growing more, and sharing them with their colleagues. Fast forward 75 years later, and we still grow what have now come to be called HeLa cells. There is likely more cell mass of HeLa cells now than there was when Henrietta was walking around all those years ago. Thanks to a successful court case, her family is now compensated for the use of the cells, and there's a lot of paperwork to get access to them. They're very broken, but they grow and spread like crazy, and so are still very easy to grow and use. Now that the field has developed, we no longer just grow cancerous cell lines. We keep healthy primary tissue alive, and even have protocols for so-called immortalization. That's where you take your cell of interest and tweak it so it'll continue to divide endlessly, instead of self-destructing after a set number of divisions as it normally would. When it comes to growing cells in the lab, that is a grade of necromancy we have become very familiar with on this channel. In the past, we've taken plant tissues, removed all of the cells, and then converted them into meat by growing meat cells on them. We made both a meat grape and a meat leaf. And for our neuron project, where we're trying to get live neurons to play video games, we maintain cultures of growing human or rat neurons. These neuron cultures, if tended properly, can last for years, and that's just a sheet of neurons grown flat. When we eventually do a video about organoids, we'll show making cultures that could last for the better part of a decade if tended well. But those examples of cells outliving their donor only exist because they're carefully tended and fed by humans, and usually they're stored at minus 196 degrees Celsius in liquid nitrogen when not in use. What about cells without any help? Well, we know that bone marrow is good for at least 5 days, and that's a tissue that's both extremely delicate and deprived of oxygen in a corpse. So what about just some muscle, or better yet, skin, which is exposed to air? If you think about it, chickens that are in the grocery store have only been dead for about a day. Same for basically everything other than beef, which is often aged in a fridge for a couple of weeks before it's sold. That would mean that every piece of fish, chicken, and pork should still be mostly alive, and maybe beef will have a little bit of life left in it too, which has some very interesting implications which we'll come back to in a moment. However, it's one thing to know that in principle it's alive, and something else to see it for yourself. So let's just test it and see. These days, this is a standard test we do in the lab all the time, and it doesn't require a source of radioactive material the way it did in the 60s. To get started, we went to the nearest grocery store and picked up the Surf and Turf special. Some pork, beef, fish, and chicken. What we're going to do is, one at a time, bring each piece of meat into our sterile hood in the biolab. The meat needs to be removed from its packaging, surface sterilized, and then a suitable piece of tissue must be removed and placed into a sterile petri dish. No sense going through all this effort just to let bacteria overwhelm the experiment. We took samples from everything: chicken skin, muscle, marrow, and tendon, various parts of the fish, and a few samples from both the steak and pork. These different samples were transferred to a 12-well culture dish, and first they were rinsed with a sterile saline solution called HBSS. This is to wash away any of the harmful products that would have built up from this being, well, a corpse. Then the samples are covered in a growth media called DMEM, supplemented with 10% FBS. We've used this mix many times before, and it's the basic recipe that most cell types can be grown in. We then transfer the tissue samples into our meat cubator to grow for a day and wake back up. The next day, we can use what's called a live dead stain to see what's what. The way this works is very simple. There are two dyes. In their base state, neither of them are fluorescent. The first one, the live stain, can penetrate into a cell, at which point enzymes inside the cell will activate it, converting it to a bright glowing green. If the cell is alive, the dye will be activated and stay captured inside the cell. The dead stain, on the other hand, works a little bit differently. This dye binds to DNA, at which point it starts to glow red. But it can't penetrate cell walls. So if a cell turns red, that means that its membrane has failed, allowing the dye in. Healthy cells constantly repair their membrane, so shouldn't allow any dye to reach their DNA. We did the staining in HBSS rather than DMEM to avoid any stray reactions that might have happened because of components in the FBS. The cells are allowed to incubate in the dye solution for 30 minutes, and then we can check and see how alive they are. To my absolute shock and horror, everything was alive. Like, very alive? Like, very, very alive. Sure, there was plenty of dead zones. This is grocery store meat. But we were shocked at how much of the material gave us a live signal, even after multiple media changes and times soaking in culture media. Don't get me wrong, there's a ton of dead cells in here too, but this was far more alive than I expected. We even got bright green signals from some little pieces of the beef, which was shocking as that has been dead by far the longest. So Mutilda's odds are suddenly looking a lot better. This does mean, though, that your sushi and steak tartare is alive when you eat it. Same if you have your steak rare. Now, while the thought of eating something alive may seem odd, you've done that every time you've had fresh fruit or vegetables. With the exception of pickled or processed vegetables, the point where they start to rot is also the point where they died. Which means that whole head of lettuce or those tomatoes are alive when you eat that salad, and the apples are alive when you bite them. So yes, even the vegans get to be horrified by this video too. Anyway, let's now talk a little bit about Necromancy Level 2, and what we can do with the cells if we catch them before they all perish. Part of the reason we decided to make this video and do this test is because we use cells in our work all the time, but buying prepared cell lines is really expensive for the tiny amount of tissue you get, so it's extremely limiting. Our thinking was that if we can use grocery store meat as the source of our cells, first off, we don't need to work with live animals, which is a huge boon. And if we need a specific tissue type, it's pretty easy to just buy a bit of brain or a kidney or whatever else as cheap offal that often just goes to waste anyway. A pack of chicken hearts is maybe $4, but would give us enough material to make a small army of meat robots. So the obvious question is, can we establish a healthy growing culture from these samples? Unfortunately, I'm just going to have to jump to the ending of that question because the answer is both yes and no. Can it be done? Yes. Did we manage it? No. But the reason isn't the source of cells, we think. There's something wrong with our protocol. The reason I say that is because after having it fail a bunch of times with the grocery store samples, we did get a few rats to try with off-screen, and those were very much alive when we got them, yet we still didn't get a good culture. So if you do cell culture professionally, either send us an email with your favorite tissue isolation protocol, or just leave it in the comments. We figure we're either timing something incorrectly or using the wrong digestion solution in the protocol. So we'll come back to getting the cell culture established in a future video, but I have full confidence that it can be done seeing these results. Once we do that though, it opens up a lot of possibilities for our long-term ongoing projects. Be it the meat robot or the neuron project, having a dirt cheap source of cells makes testing out our ideas so much easier. And in combination with our past video replacing culture media with Gatorade and household FBS substitutes, we've almost got it to the point where growing animal tissue will be just as easy as growing bacteria or fungi. But those are just the current projects that can be sped up. What else could we do with a culture of cells harvested from the grocery store? In the universe of Dune, one of my all-time favorite series, there's a delineation between clones and golas. A clone is Well, it's a clone. It's a copy of somebody made from a sample of living tissue. A Gola, on the other hand, is a clone made from material taken from a corpse. I've always wanted to try cloning something, and behind the scenes we've begun working to see if we could clone a chicken. They're readily available, the eggs are huge, so in theory they're easy to work with, and they take up a lot less space than a cow. Sorry, Mutilda. But the same process, in theory, works on any animal. Once we have an established cell line from a grocery store chicken, we can do what is called somatic cell nuclear transfer. That's the proper name for what has come to be called cloning. Basically, you take an egg cell and a cell from your established culture and do a little switchy-switchy with the nucleus of the cells, moving the source chicken's nucleus into the egg cell. And then you literally zap it with electricity to get it to start replicating and growing, which I personally find extremely weird that works. Now, the produced animal wouldn't have the mind or memories of the original one, but somewhere in a field someday will be Hennifer 2 Electric Boogaloo, clucking away and eating bugs. And then the cycle can begin anew. Though that does beg the question, is it more ethical to eat many different chickens, or just copies of the same one bird? I honestly don't know.
Yan. Good.
However, if you take the resulting birds and cook them, it's now crossed the next threshold, after which is... Death number 3 is your physical remains, and most importantly, samples of your DNA. Even after every cell in your body is dead, there's still a little bit of you left. Your skeleton and whatever other bits of tissue remain carry what's left of your DNA, and a history of your life. And while wear and tear on your teeth can tell a future archaeologist about your terrible eating habits, it's the remnants of your DNA that we're most interested in for the sake of today's discussion. So, how long does your DNA stick around, and at what point is it no longer possible to pull a Jurassic Park? We didn't get a chance to talk about it when we made our Egyptian mummy, but something we came across in our reading while preparing for that project was that Egyptian mummies were recently sequenced thanks to DNA left over in their teeth. And even over longer time spans, some traces remain. We've managed to get DNA out of some truly ancient fossils. This is how we know that some of our DNA is from Neanderthals, for example, because we were able to sequence the incredibly old Neanderthal fossils and then compare them with samples from modern-day humans. Even in dinosaur fossils, some traces of soft tissues and protein can remain even after millions of years. There is a reason I bought this piece of T-Rex bone, after all. A group of researchers subjected T-Rex fossils to mass spec analysis, and they were able to piece together the amino acid sequence of the tiny amount of highly degraded collagen that was left in the fossils. And, to my immense amusement, the sequence was essentially identical to that of modern chickens. But exploring that is for a future video. While it may not seem like much, even these vestiges, which are by all definitions dead, may yet give rise to life. These days, not only are we developing tools to quickly analyze and sequence whole genomes, we're also developing the tools to print out whole chromosomes. There are already many examples of researchers printing a chromosome and then switching it into a living organism. So what's to stop them from doing that with an entire organism's genome, and then loading it into an egg cell? A Gola need not be made from living tissue if you've got a complete gene sequence. Just print their DNA back out, slap it into a healthy cell, and out pops Hennifer 3 digital nomad. However, these fossils and specimens require perfect preservation, whether that was by being carefully mummified and stored in an incredibly dry place, or a bone left in a cave somewhere for 1,000,000 years. But the trouble is that most individuals of a species don't have this luxury. If your body is allowed to decay naturally, or is consumed or condemned to the depths, nothing will remain physically, and all that's left is... This one is pretty straightforward. This is your legacy. The ripples you make in time, and importantly, the amount of time that you're remembered. When even your DNA is gone and no samples remain, all that's left is the impact that you made on the world. This is the forest you helped sow, the books you wrote, and the friends and family that keep a version of you in their heads. But even these ripples are no match to the onslaught of time. Unless you're one of the very lucky, or unfortunately, often one of the very evil, time will forget you. Most people aren't Marcus Aurelius and have their books published thousands of years after their death. Or be like Genghis Khan that killed so many millions of people that there was a detectable drop in Earth's temperature from the sudden reduction in CO2 output. Though sometimes you do get lucky and happen to be a spectacularly bad copper merchant and live on in infamy because some archaeologist found your receipts a millennia or two later. Most people, though, are Ted from accounting that collects plastic ducks and plays squash with their neighbor. Or Martha, the nurse that saves lives all day and then goes home to build intricate models out of Q-tips as she downs a pint of margarita to drown out the memory of the morning's horrors. Or any of a billion other unique, interesting, wonderful, and bafflingly weird humans that will perish and be forgotten barely a few years after they're gone. Even if it takes 1,000 years, the universe is unfathomably old and isn't in a rush. That said, the universe doesn't always win the battle every time, and humans are clever little monkeys, so other than being unbelievably violent bastards, we have found a few ways to cast our memories, even if blurry, into the far future. Religion is of course one option. Tell enough people that you are God, or God adjacent, and have them copy a book or two about your life. Suddenly, a carpenter from the desert or a prince or pauper can have their stories told for thousands of years. But you don't need to look hard to find the remains of dead gods. Gods die when people stop believing in them and move on to making up new gods. You need look only as far as the pyramids of Egypt, or the Aztecs, the ruins of Greek temples, or the Easter Island heads to find the fragments of gods nobody follows anymore. All over the world, gods have come and gone, and their corpses litter the land. Though that does bring up one long-lived way to be remembered. Make a big, cool pile of rocks. It may sound silly, but take a material that doesn't weather quickly and make a huge pile of it that's decorative, and it'll probably be there 4,000 or 5,000 years later. It might get buried, but even the ancient Egyptians had archaeologists studying the ancient Egyptians. Humans love finding a cool pile of rocks that somebody else made a long time ago. But a rock, no matter how hard, will yield to nature eventually. Water may not seem abrasive, but give it a few thousand years and suddenly the world looks like it's been marred by a giant version of the water jet channel, deep gorges and valleys forming from its passage. So it is the humble opinion of this biologist that the best way to project yourself into the future is plants. Sow a forest large enough and it'll outlast the mountains. Or do something to modify a plant to be more delicious and people will still be growing that plant in 1,000,000 years. Take the humble avocado, for example. Avocados exist because they co-evolved with giant ground sloths. This symbiotic relationship shaped the taste, size, and texture of the plant, but the sloths are now long gone. In their stead, shiny monkeys, aka humans, found them tasty, and there are now more avocado plants than there ever were when the sloths were around. When the colonialists came to the Americas, they were surprised at how much of the wildlife was edible. But North America wasn't turned into a food forest by chance, it was through deliberate tending by the natives, spreading food plant seeds far and wide and making walkable food trails all over the continent. There are still vestiges of that forest left, but it's largely been paved and plowed. Down in South America, big chunks of the Amazon are the same, filled with edible plants cultivated over millennia by humans. And some areas have deep black soil called terra preta, which was made over centuries by humans, and is some of the most fertile soil on Earth. Those humans are long gone, but their life's work will be there for many millennia more, nurturing new life. So, as the old saying goes, the wisest will plant a tree knowing they will never sit in its shade. Ironically, the final level of necromancy is to live well. Make a big pile of rocks with your friends, plant some trees, write a book, keep notes on your terrible copper deals, make up new gods, and tell each other stories. Because if you do, that last death may not come so soon, and you might just survive.